Hi, my name is Gayatri Sabrina with the metric number of D19A0009. Today I will be presenting about ciliates infestation. Okay, firstly, what is ciliates? Ciliates is a group of protozoan characterized by the presence of hair-like organelles called as cilia, variously used in swimming, crawling, attachment, feeding, and sensation. The agent involved would be Ictiopitheris multifilus, which is an obligate protozoan, and it is classified within the phylum Ciliophora. The host would usually be freshwater fish such as tilapia. The species is Oreochromis nilotus, and other example would be common carp. The species would be Cyprinus carpio. For the distribution, it is common and almost found in everywhere when there is water. And for the clinical sign, we can see white spot on the fish surface that can cause white spot disease due to the proliferating epidermal cells enclosing the trophin. And also other clinical sign includes irregular swimming pattern, rubbing against the object, flashing and jumping due to skin reaction around the developing trophin. Moving on to the diagnosis, we can do evaluation of fresh wax smears from scrapings of the skin, fin and gills under a light microscope, and we can observe a horseshoe-shaped macronucleus and movement of cilia. And upon histological section, the parasite can be seen with characteristic of nucleus, hyperplasia and immune cells around the trophin. And for the complementary diagnosis, PCR and qPCR can be done. For the vaccination, there is no effective commercial vaccines available for ciliates in fish. For the life cycle, firstly, the first stage will be trophin, which is internal in the fish, while the others are external, with the multiplication step occurring in the water. So firstly, the mature trophin exit the fish surface and transform into tomen, which is a free swimming stage that seeks surface of submerged elements and fish tank walls for encysement. The tomen then produces a gelatinous cyst wall and thereby transform into tomocyst stage. And then asexual reproduction takes place in these cysts by numerous mitotic division. Then the resulting tomites move strongly within the cyst and will eventually escape through the cyst wall. The free-swimming tomites are termed as pterons and will seek a fish host and penetrate the epidermis or gill epithelium in order to transform into the trophin stage, which exhibit temperature-dependent growth. And when the trophin reach a size of 0.2 to 1.0 mm, it will leave the fish host by disrupting the epidermis and gill epithelium. For the transmission, the infective pteron stage of the parasite invades the skin and gills of the fish, which then penetrates the epidermis and settle above the basal lamina. And here it actually transforms into the trophin stage that feed on fish tissue until it reaches a size of 0.2 to 1.0 mm, which is macroscopically visible as a white spot. The trophin will then constantly move within its location on the fish surface, which is the skin, gills, and the fin, while it ingests the cells and cellular debris, which are transported by cilia to the cytosol. So here high mortality occurs and the fish may die either from penetration of the numerous pterons into the skin or gills, or when there is numerous of trophin escape from the fish surface. Last but not least, the predisposing factor would be poor water quality, high stocking density, stress due to handling, and weak immune system or concurrent infection. That's all from me. Thank you.